We are going to talk about a little bit about antigen presentation. The antigen presentation is in the innate system. It can be activated by the free antigens, but the adaptive system requires antigen presentation. Adaptive have to require antigen presentation by the antigen presenting cells, etc. And that pathogen must be engulfed by the cells, broken down, then that pieces of proteins are transported to surfaces and that antigen presented to T cell for activation. That is happening in adaptive immune system. But in innate immunity, the free antigens also enough for the activation of innate system. The second, there is cytokines. The cytokines are cell signaling proteins. Cell signaling proteins, and they are often released by immune cells and they stimulate the inflammatory responses in various subsets. They are having different functions like chemokine. The chemokine means they will attract immune cells. They processes cause chemotaxis. They are having interleukins. Interleukin 1, 2, etc. can tumor necrosis factor and then can cause the tumor death. We are also having transforming growth factor. The transforming growth factor also we have interferon. Why we said interferon? Because they interfere with viral replication. And most of this you guys study like CD. And CD means cluster of differentiation. Cluster of differentiation. These cluster of surface differentiations are several surface molecules like CD3, CD4 and CD8 etc. and found on many immune cells like T cell and B cell used to identify cell type. Some used as receptor cell binding. So we are also having innate immune system in which there is a phagocytosis, and in phagocytosis the macrophages are hallmark, and hallmarks are also for that reason you have a neutrophils there. We have in complement, natural killer cell, eosinophil, mast cell, and basophil, already you guys know. And also they recognize molecules that are foreign, they having the pathogen associating molecular patterns, and also they use the pattern recognition receptors and key receptor class in the innate immunity is toll-like receptor. The macrophages, dendritic cell and mast cells, and they recognize the PAMs and secrete cytokines. In innate immunity, you guys already know about all the key features for the pathogen mechanism, but we have to know about the macrophages. The macrophages are monocytes. Macrophages are guardians of innate immunity. They produced in bone marrow as monocytes. Next, monocytes. In the bone marrow, they are present as monocytes and they circulate in blood for 3 days. And when they are entering in tissue, these monocytes become macrophages. And given, according to site of macrophages, you are giving them a name like FasL, that are present in liver, they have in the microglia, that are present in CNS, they are osteoclasts, that are present in bone. Yes. And they are having 3 key functions. The first is phagocytosis. The first is phagocytosis, second is cytokine production. The macrophages also by producing cytokine increase stimulated T cells, and they also have antigen presentation. These are three basic and important functions of macrophages. And how they will go for the phagocytosis? The macrophages engulf pathogen into phagosome, and the phagosome merge with lysosome. And these lysosomes contain deadly enzyme, and that causes death of bacteria and viruses. And they are having the reactive oxygen species like superoxides, and that produced by NADPH oxidase. If you guys remember about respiratory bursts, and they generate the hydrogen peroxide and oxygen radicals. And the active nitrogen intermediate also used for killing of bacteria. Like for example, there was a nitric oxide with superoxide that will make a combined combination and form the peroxynitrate. Also a strong compound against bacteria. And we use different enzymes like proteases, nucleases, and lysosomes, and enzymes, and that hydrolyzed peptidoglycan glycan. Secondly, some pathogens block this process like phagocytosis. It is not like that our system are going to overcome all the bacteria, but some pathogens are going to block phagocytosis process, and that is mainly by the tuberculosis. The tuberculosis modify the phagosome, and they will be unable to fuse with lysosome. When the phagosome and lysosome did not fuse, then there will be not proceed the function, the process will not proceed for the destruction of bacteria, and there will not proliferation inside the macrophages and protection from antibodies. So if you guys know about the Shedick-Galschi syndrome, someone these days syndrome, there is the immune deficiency syndrome, and there are a failure for lysosome to fuse with phagosome, and this disease is a recurrent bacterial infection. What is main characteristic? This disease, a lysosome is unable to fuse with phagosome. And by that process, they are not able to kill the bacteria, and there will be recurrent infection. The macrophage can exist in several states. It is interesting. The macrophage is doing tablets removal, but in the activated form they are more active. They are more active, and the major activators are via toll-like receptor. Look at all again, toll-like receptors, and in which they will lipopolysaccharide from bacteria, peptidoglycan, bacteria DNA that are not methylated, and also interferon gamma from T cells and natural killer cells. They also stimulate, and the macrophages, the interferon gamma is going to attract or activate the macrophages, and it is attracted by the gamma to move to migration macrophages by complement system, including C5A. That is so important points. That are so important points. The interferon gamma is going to stimulate macrophages, and that are attracted by C5A. C5A. Okay. we're having different surface markers and signals. For example, the key surface receptor in the macrophages we're having. The key surface markers, like if there's a LPS, lipopolysaccharide from a gram-negative, it will going to attach with CD40. If there's a FC antibodies, then they are going to attach with FC receptors. If there is a C3b complement, they are going to attach with C3b receptor. There are some T-cell molecules like CD4, they are going to attach with MSH. MSH2. And if there is CD28, they are going to attach with B7. If they are CD40 ligand, then they are going to attach with CD14 present on macrophages. That is so important. That's why you have to remember for upcoming topics that CD4 is going to attach with MSC class 2 and CD28 with B7 and CD40 ligand with CD40. And guys, the macrophages, the key cytokines are produced by macrophages are interleukin 1 and tumor necrotic factor alpha. These are key cytokines of macrophages, and other are interleukin 6, 8 and 12. The interleukin 1 and tumor necrotic factor alpha both are increased in synthesis endothelialization molecules and allow the neutrophil to enter the inflamed tissues. The interleukin 1 is endogenous pyrogen that causes a fever. And secondly, it acts on the hypothalamus. The tumor necrotic factor alpha can cause a vascular leak in the septic shock, and there is a catalytic inhibit lipopolyprotein lipase in the fat tissues, and it reduces the utilization of fatty acids. This is causes cachexia and it kills tumors in animals. Therefore, we are saying tumor necrosis factor, and then cause intravascular coagulation DIC. So, in tumor necrosis alpha, there is a high chance of DIC disseminated intracoagulopathy.